Welcome to HyperAcademics Neuroscience 2 video tutorial 2 on basal ganglion. The basal ganglion is a mass of gray matter at the base of the cerebral hemisphere. The basal ganglion, while it does not have any known direct connection with the spinal cord, it is known to have an influence on the corticospinal tract, and thus it is said to have control over movement. Additionally, in addition to its involvement in the execution of movement, it, alongside the cerebellum, is also involved in the storage of motor plans. The basal ganglion is divided into two classifications, the traditional and the clinical classification. In both classifications, you will find consisting in there the lentiform nuclei and the caudate nuclei. However, unique to the traditional classification, you will find the claustrum and the amygdaloid body. And unique to the clinical classification, you will find the subthalamic nucleus and the substantia nigra. The lentiform nucleus, which forms part of the basal ganglion, is divided into two parts, one of which is divided into two parts further. So essentially, the lentiform nucleus has three parts. On the most external aspect, or the most lateral aspect of the lentiform nucleus, you will find the putamen. Medial to that, you will find the globus pallidus, which is divided into two parts, the globus pallidus externa, which you see here, and the globus pallidus interna, which you see here. You will definitely hear about and certainly need to remember these terms: the corpus striatum, which includes the caudate nucleus and the lentiform nucleus; the neostriatum, which includes the caudate nucleus and part of the lentiform nucleus, being the most external aspect, which is the putamen. And then you will hear and need to know the paleostriatum, which is the globus pallidus, both interna and externa. I implore you to remember these facts, as these things are things that can come in the multiple choice questions. Now, the fundamental thing about basal ganglion that you need to know is the fact that there is what is known as the putamen circuit, and there are two pathways, the direct and the indirect pathways. Now, before we go into the intricacy of the pathways, you need to know the fibers that are involved. You also need to know where the fibers are coming from and where they are heading to. And probably most importantly, you need to know what is the neurotransmitter involved. So we start. From the cerebral cortex to the putamen of the lentiform nucleus, you are going to find corticostriatal fibers. These fibers are glutaminergic fibers, which means they release the neurotransmitter glutamine. These fibers are stimulatory fibers. Next, you're going to find the striatopallidal fibers. These fibers come from the putamen to the globus pallidus. And these fibers are inhibitory fibers. They release either GABA or substance P. Next, you have fibers coming from the globus pallidus to the thalamus, these known as the pallidothalamic fibers. And these fibers are also inhibitory fibers, releasing GABA and substance P. And last but by no means least, you have the fibers that reconnect the circuit to the cerebral cortex from the thalamus. being known as the thalamocortical fibers. And these fibers are glutaminergic, just like the corticostriatal fibers. So what is the neurotransmitter released? That's right, glutamate. So now that we know the fibers, where they are coming from and where they are going, and the neurotransmitter that is released, let us, as the Jamaicans say, maths it out a little bit. Once you have this fundamental principle of what are the fibers, where they are coming from and heading to, and the neurotransmitter released, then the entire basal ganglion business becomes very simple. So look at it. Always think of it this way, that what we are involved in is motor, movement. In order for movement to take place, the cerebral cortex has to be stimulated some way. But what happens is that in the absence of any stimulation in the lentiform nucleus, the fibers from the thalamus, which are responsible for stimulating the motor areas of the brain, like the premotor area, the supplementary cortices, and the frontal eye field, etc. These fibers, when there is no stimulation to the lentiform nucleus, particularly the globus pallidus, these fibers are forever inhibited. So they are inhibited by GABA, which is released from pallidothalamic fibers. But let us take it from the very beginning. So fibers from the cerebral cortex to the putamen, also known as corticostriatal fibers, are glutaminergic fibers. They release glutamate, and so they are going to stimulate the fibers that are next in line, known as the striatopallidal fibers. By stimulating the striatopallidal fibers, which themselves are fibers that are inhibitory, what this does is that it stimulates the release of the inhibitory neurotransmitter, which is GABA. And this GABA is going to inhibit the next fiber in line, which is the pallidothalamic fibers. These fibers are what, under no stimulation, release GABA to inhibit the thalamocortical fibers. So when it is that the striatopallidal fibers are stimulated, GABA is released in the globus pallidus internus, and that is going to inhibit the pallidothalamic fibers. And thus, by inhibiting the inhibitory fibers, the net result is going to be stimulation. If you release or remove the inhibitory stimulus, then what is going to happen is stimulation. So by inhibiting the pallidothalamic fibers, what happens is that the thalamocortical fibers are no longer under the influence of GABA, and thus it is able to fire, releasing glutamate in the respective areas of the cerebral cortex, and thus facilitating movement. It is important to note that the thalamus acts as a relay station, and it is related functionally and intimately with the cerebellum, and therefore computation occurs in the thalamus. And that basically, students, is the direct pathway. Know it at the back of your hand. The three things you need to know: what are the fibers, where they are coming from, where they are heading to, and what is the neurotransmitter released at the various fibers. Now, remember, for movement to occur, there must be simultaneous agonizing and antagonizing. So if you are to move your forearm up and down, you're going to need to stimulate extensors and inhibit flexors, and in the opposite movement, inhibit the extensors and stimulate the flexors. So in order to do that, seeing that we know the direct pathway is a stimulatory pathway, we need a pathway that is going to be involved in inhibition, and that's where we come to the indirect pathway. Now, the indirect pathway is a little different from the direct pathway. I apologize. On this slide, it should read indirect pathway. I repeat, on this slide, it should read indirect pathway. The indirect pathway involves the subthalamic nucleus. So it involves the three parts, the cerebral cortex, the lentiform nucleus, and the thalamus, which is just like the direct pathway. But the indirect pathway, as I repeat again, this should be indirect pathway, also includes the subthalamic nucleus. So basically, what you need in this indirect pathway is you need the thalamocortical fibers to be inhibited, because what you're trying to do is antagonize the action, the stimulatory action of the direct pathway. And so just like the direct pathway, you have glutaminergic fibers coming from the cerebral cortex. However, these fibers act on special fibers coming from the putamen to the globus pallidus externa. The second fibers in the direct pathway would have been from the putamen to the globus pallidus internus. But you need to know the first difference between, or rather the second difference between the direct and the indirect pathway. The first being that the direct is a stimulatory pathway and the indirect is an inhibitory pathway. The second is that the second set of fibers, or the fibers from the putamen, known as the striatopallidal fibers, come from the putamen to the globus pallidus externus in the indirect pathway, and in the direct pathway from the putamen to the globus pallidus interna. So the corticostriatal fibers are going to stimulate these fibers from the putamen to the globus pallidus externa. And what that does is it stimulates the release of GABA or substance P, both being inhibitory neurotransmitters. And these inhibitory neurotransmitters in the globus pallidus externa, these neurotransmitters are going to inhibit the release of inhibitory neurotransmitter in the subthalamic nucleus. So understand that when there is no stimulation, there is release of GABA in the subthalamic nucleus. But once there is stimulation, GABA released in the globus pallidus externa is going to affect the fibers from the globus pallidus externa to the subthalamic nucleus, and cause inhibition. This inhibition of the inhibitory fiber means that the constant inhibition that would ordinarily take place is going to be itself removed or inhibited. And thus, fibers from the subthalamic nucleus now going to the globus pallidus internus are going to be stimulated. These stimulation taking place in the globus pallidus internus by action of glutamic neurotransmitter is going to stimulate the release of GABA in the thalamus, and thus the release of GABA is going to inhibit the thalamocortical fibers, and thus inhibition occurs in the cerebral cortex. So to quickly go through it again, the corticostriatal fibers in the indirect pathway is going to release glutamine, which itself is going to stimulate the release of GABA in the globus pallidus externa, which itself is going to inhibit the constant release of GABA in the subthalamic nucleus. and that is going to release the fibers from the subthalamic nucleus to the globus pallidus internus, release it from any inhibition, and thus result in itself being stimulated to release glutamine in the globus pallidus interna, and thus that stimulation is going to stimulate the pallidothalamic fibers which run from the globus pallidus internus to the thalamus, and thus by stimulating this inhibitory fiber, it's going to result in the release of GABA in the thalamic nucleus, and thus inhibition of the thalamocortical fibers. Again, you need to know what are the fibers, where they are coming from and where they are heading to, and the neurotransmitters involved, and understand the difference between the direct and the indirect pathway. In the direct pathway, there are fibers running directly from the putamen to the globus pallidus internus. The direct pathway is a stimulatory pathway. However, in the indirect pathway, which you are seeing before you, again, please ignore the fact that there is this error. In the indirect pathway, the fibers from the putamen go to the globus pallidus externa, and these are going to inhibit fibers to the subthalamic nucleus, which is going to cause a stimulatory sensation from the subthalamic nucleus to the globus pallidus internus, and then that is going to stimulate the release of GABA in the thalamic nuclei, and then you're going to have a net inhibition of whatever center, motor center in the cortex. We have to also understand as well that the direct pathway is much shorter than the indirect pathway, and it activates muscles. However, the indirect pathway is much longer, and it deactivates muscles. Now, right here, what we have is the substantia nigra. The substantia nigra has two parts. The parts that we are interested in is in the pars compacta. Why are we interested in the pars compacta? The pars compacta release a substance known as dopamine via nigro or nigrostriatal fibers. That is fibers running from the substantia nigra, particularly the pars compacta, to the putamen. So the release of dopamine takes place in both pathways via these nigrostriatal fibers. The function of this dopamine is to basically kickstart or initiate the excitatory or inhibitory pathways. So it stimulates both the direct and the indirect putamen circuit. There are fibers, special fibers coming from the cerebral cortex, which you won't see demonstrated in this diagram. These fibers are known as corticonigral fibers, and they relay information to the substantia nigra to let it know what is happening. Now, the role of dopamine. Glutaminergic and dopaminergic releases are antagonistic to each other. That is, one antagonizes the other. It is the change in this balance that determines the net action. If it is you have a greater influence of glutamine, which is stimulatory, then the net action will be stimulatory. If it is that you have an imbalance in favor of dopamine release, then the net result is going to be inhibitory. Now, there are two receptors, two dopamine receptors, one known as D1 receptors and the second one known as D2 receptors. Dopamine is going to have a different action based on which of these receptors it binds to. If it binds to the D1 receptors, usually found in the direct pathway, then what is going to happen is stimulation. And if it is that you have the D2 receptors in the indirect pathway being stimulated, then you're going to have an inhibition of the indirect pathway. Okay? The other aspect or the other part of the substantia nigra is known as the pars reticularis, and this is known to be to have similar function to the globus pallidus interna. So it likely has connection between itself and the thalamus. Within the striatum also there are what we know as cholinergic neurons. Acetylcholine is released, and this release has inhibitory effects on the direct pathway and stimulatory effects on the indirect pathway. Not so fast. We have one more thing to go through, and that is the pathologies. Now, anytime you have a lesion in the direct pathway, which is a stimulatory pathway, you're going to have what is called hypokinesia, which is a reduction in motor activity. It makes sense that if it is that the direct pathway causes increase or it facilitates motor activity, a lesion in that pathway is going to cause the opposite, which is a reduction in the ability to facilitate movements. Likewise, if you have a lesion in the indirect pathway, what you're going to have is called hyperkinesia or dyskinesia, because the indirect pathway is supposed to maintain a certain level of movement that is minimal. It is supposed to inhibit movement. So if you have a lesion, an abnormal lesion, then the opposite is going to take place, and you're going to have an increase in the movement that takes place. Further to that, it is important to know that if you have a lesion in the substantia nigra, you're going to have a Parkinson-like disorder, which may involve or may include rather pill rolling syndrome, resting tremor, bradykinesia, stooped posture, etc. So just know that if you have a lesion in the substantia nigra, you're going to get a Parkinson-like disorder. Additionally, if you have a lesion in the caudate nucleus or the neostriatum, you're going to come down with a chorea, such as Huntington's chorea, which mainly affects males, family-linked, and normally present after the age of 40. If you have a lesion in the subthalamic nucleus, you will get ballism, which is that spurt of movement, ballistic kind of movement of the limbs, which itself can be very violent at times. And then you have the neostriatum. If it is you have a lesion in the neostriatum, you're going to get what is known as athetosis, A-T-H-E-T-O-S-I-S. It is not really known what is the big difference between the chorea and the athetosis in terms of the cause, but just know that once you hear caudate nucleus, the pathology due to a lesion is known as the chorea, and in neostriatum, it is athetosis. And now, here ends the basal ganglia neuroscience 2 tutorial of HyperAcademics. Thank you very much for joining us, and do join us some other time.